This is a second video of the synthesis and pre-mRNA cap formation. We understand the promoter has a core promoter element, tata box and initiator region, and enhancer. It means promoter has core promoter and enhancer. The core promoter has tata box and initiator. Okay, you can summarize for yourself like a tree branch. This one and this one, this one. Understand me? Right?
But
everything like this.
Does the promoter have an enhancer element?
Promoter, yes. You see, all of this structure when it's back here,
But
all of them are promoter. This is a core promoter. This is a
what is in the core promoter?
This is the enhancer element. Enhancer element of the promoter, core promoter. But both of these photo is promoter.
Oh, the whole thing.
Whole thing.
I only thought that only this thing down.
No, this is a
down
core
there.
promoter, and core promoter has a tata box and initiator region. Now,
What is the difference? Like the initiation, like the
initiation
and the tata box?
is the element of the DNA. The RNA polymerase is bound to.
Yeah.
The tata box is the place for the tata binding protein and all the transcription factor and all of the activator and coactivator.
Okay.
The binding place is different.
Okay.
We have a RNA polymerase 2 core. RNA polymerase 2 has a core polymerase, initiator factor, and this is a holo enzyme. And why this is holo enzyme? This is not, the photo is not here again. And see, I don't have the good photo of that also here, but the RNA polymerase 2 is like this exactly. Okay, this is a mRNA, for example, and this is a core polymerase. Core polymerase. The initiator factor should come here. Initiating factor IF, and both together is holo enzyme. Always the holo enzyme is a big enzyme. Have a lot of job.
But what is the difference in core
Core is a center, is a nucleus of this protein. For example, this is a very big protein,
Yeah.
and the match here is a core of that. This is a core, and all of the finger is an initiator factor, and the job of that is transcribe the mRNA. Also, RNA polymerase 2 has a histone acetyltransferase, has a helicase subunit. The helicase subunit is here. Helicase subunit. For example, one of the alpha or beta or delta, you learn it next semester in the biochemistry. One of them is do the histone acetyltransferase. It has also C-terminal domain, CTD, of a 1 polymerase 2 subunit, consists of heptapeptide repeat, bonds strongly to mediator, and phosphorylation of C-terminal polymerase fucking
Because I'm not gonna remember all of this. Like, what is the most important?
But if you remember this photo,
Only
I
this-
show it, initiator factor, core
Yeah.
polymerase,
These two makes the holo enzyme. Was it?
all of them is holo enzyme.
Oh,
all
okay.
of them. See, plus together is equal to holo enzyme. This
But-
is the, we have a lot of holo enzyme. Holo
Okay.
enzyme is a big enzyme. No, RNA polymerase 2 is one of the holo enzyme.
Okay.
Okay, it means it has a core and it's somebody help it, and also the at the extra around the core we have a lot of different enzymes like a helicase, like an acetyltransferase, like a CTD, and CTD is one of the polymerase 2 subunits. You see, I should show it to you the photo and you understand it very well. See, this is a RNA polymerase 2. This is a CTD. This is a C terminal domain here. Okay, the initiator factor, transcription factor bonds to RNA polymerase. The other thing, for example, on the behind is helicase subunit and such like that. But we should have one very nice more photo of that. These are the different species.
But what is the core promoter elements like? But that is.
Core promoter is this one. It's a big one.
Is a big one.
It's a big one.
Then where's the
nucleus
RNA polymerase in this cell?
This is a RNA polymerase, and this is a core of that. This is a C-terminal domain and initiator factor. For example, one of these polypeptide do the helicase subunit, the other polypeptide do the acetyltransferase, the big enzyme. We have a lot of holoenzyme in the body, and because they are a big enzyme, they are the complex of everything together, do a lot of job. For example, one of the job is histone acetyltransferase, the other job is a C-terminal domain, and C-terminal domain consists of the heptopeptide bonds to strongly mediator. This is a job of C-terminal domain, bonds strongly to mediator, and phosphorylation of CTD, release of mediator happens. Because of the lot job, this is a place of mutation. And if the mutation happens, maybe the mRNA is overproduced, or maybe the lack of the mRNA produced, or lack of the protein production. And because of that, that's very important later in the drug therapy treatment of the disease. Okay. Again, what is the RNA polymerase 2? Holoenzyme. Holoenzyme has a core polymerase, has a helicase subunit, has a histone acetyltransferase, and CTD. The CTD, what is the job of CTD? Bonds strongly to mediator. What is the mediator? For example, here, when it's bound to here, this is the start of the transcription. And here is written here acetylase, the acetylase, and they are, for example, these are also the part of the RNA polymerase. But this hand, for example, is here, histone acetyltransferase. Understand? The all together, this complex together is RNA polymerase 2. And if the CTD is phosphorylate, the polymerase 2 release from mediator and transcription is a start.
What is the mediator?
You see, it's written bonds strongly to mediator and phosphorylation of CTD, polymerase 2 release from mediator and transcription start. It means mediator is something can stop the transcription. Because if the CTD is a phosphorylate, if the CTD, the big one, is phosphorylate, it releases from RNA polymerase 2, and RNA polymerase 2 can start the transcription.